Good morning. I am Rachita Kumar, an alumnus of Shastra University, India, and I will be talking to you on the systems modeling of the skin microbiome. This project was carried out with the guidance of my mentor, Dr. Karthik Raman, at the Computational Systems Biology Lab in the Indian Institute of Technology, Madras, India, for my undergraduate thesis. Microbial communities are driven by various organisms, and their compositions are influenced by variations in environmental factors such as diet and physicochemical characteristics such as pH, temperature, salt concentration, etc. Here in the skin microbiome, we primarily have 4 skin sites, namely dry, moist, sebaceous, and foot. These sites are colonized by a wide range of eukarya, viruses, and bacterial species primarily from actinobacteria, proteobacteria, firmicutes, and bacteroidetes phyla. Their interactions within the community and those between the community and the host have major implications in several skin diseases like acne, dermatitis, leprosy, psoriasis, etc. Microbial interactions prevail in these communities, and there are many ways to model these interactions. Our primary objective was to try to unravel the complexity of these interactions through metabolic modeling, and for this we used a graph-based approach. Our approach was threefold. First, to study the metabolic interactions between the species. Second, to construct a microbial association network for the microbiome. And third, to understand the specific mechanisms through which species interact with each other, support each other, or depend on each other. For our analyses, we used the available genome-scale metabolic models for the bacterial species in the community from the VMH Agora 1.01 database. To analyze the metabolic networks, we employed MetQuest, an algorithm previously developed in our lab. It is a graph-based method to enumerate metabolic pathways of a specified size from a given source to a given target metabolite. We employed this to traverse the bipartite graphs of these metabolic networks and understand how well one organism supports another in terms of relieving the stuck reactions. A stuck or blocked reaction is one that cannot proceed due to the lack of precursor metabolites in the individual organism. These may be supplied by other organisms in the community or by the environment. This support is quantified by the metabolic support index given by the fraction of blocked or stuck reactions relieved in the presence of another organism in the community. From our analyses, we found that Corynebacteria are highly supportive species in the community, generating high MSI values in their presence. It appears that Corynebacteria require little support from the community, offering little or no support amongst themselves. Staphylococci are highly dependent species and rely on Corynebacteria and Streptococci for support. Veillonella parvola is the most dependent species in our dataset with a high MSI value of 0.39. Interestingly, comparison on a strain level in our dataset reveals that 2 strains of Staphylococcus capitis metabolically support the community differently. Probing into their interactions with a highly dependent species, Veillonella parvola, we observed that while one strain offers little support, with a low MSI value of 0.01, the other strain is highly supportive, resulting in a high MSI value of 0.38 in its presence. We further constructed a microbial association network using the MSI values. Here we see that Corynebacterium tuberculostiricum is the most supportive species in terms of the number of species that it interacts with in the community, followed by Propionibacterium acnes, Streptococcus aurelis, and other Corynebacteria. Further, we were curious to understand the different ways in which support is rendered by these species. We classified the relieved reactions into their respective enzyme classes. and on computing the average percentage of reactions relieved by each species in each of these classes, we found that many of them render support largely in transferase and translocase activity. We are eager to further study the biological implications and relevance of this and take these studies forward. To summarize, we used a graph-based approach on metabolic networks to study the skin microbiome and understand the various interactions in the community. These interactions were quantified using the metabolic support index and were used as a basis to model and represent the microbiome. Further, these interactions were probed into to determine if there were specific ways or mechanisms in which these species supported each other metabolically. Additionally, the analyses were done to identify the variations amongst genera, species, and even strains. Thank you and have a great day.